In general, DNA consists double-stranded of nucleotide sequences, and restriction enzyme is an enzyme that cuts DNA at a specific nucleotide sequence known as restriction site. So, to further our topic for today, my other four friends will explain more about this in details.
What is the restriction enzyme? Restriction enzyme is the one way that bacteria cells protect themselves from the bacterial phase is by using special protein called restriction enzyme or restriction endonuclease. This restriction enzyme can cut the viral DNA at a specific location, thereby destroying its activity. As we know, bacteria do not have the antibodies like human to protect themselves. So, to protect themselves from the bacterial phase, they use the restriction enzyme. Other way, scientists discover how the bacteria protect themselves use the restrict enzyme. Scientists was apply this application of restrict enzyme for the molecular technology.
From the study, we realized that restriction enzyme cut the specific location which called as palindromic sequence of DNA molecule. What is palindromic sequence? Let's take a look at the following diagram. Let's suppose we have this section of palindromic sequence. But first we need to remember that double-stranded of DNA consists of two single-stranded of DNA. So this double-stranded consists of complementary base pairing, which the orange color is adenine, black color is thymine, and the green color is guanine, and the blue color is cytosine. Basically, the restriction enzyme will look for this sequence of DNA and cleave the DNA molecule. So, for example, we add some restriction enzyme from this sequence. So, as a result, it will form an asymmetric cut, and as a result, it will have two single-stranded exposed, which is called as sticky ends.
Based on this picture, there are two types of bacteria, which means bacteria number 1 and bacteria number 2. These bacteria came from different sources. In the laboratory, the restrict enzyme we added into the appendage tube, where the two sources of bacteria already have in the tube. The restrict enzyme will move along the DNA sequence to find the palindromic sequence. After it finds the palindromic sequence, the restrict enzyme will cut the DNA sequence like the asymmetric manner, which is to create the sticky end, like this. So, the original DNA sequence will not bind to each other because the restrict enzyme, which means after we added the restrict enzyme into the appendage tube that have the bacteria number 1 and bacteria number 2, the restrict enzyme will find the palindromic sequence in the DNA sequence. After it finds that sequence, it will bind to the restrict enzyme. So the restrict enzyme will create the asymmetry manner. So the original DNA sequence will not bind to each other. The DNA form of the double helix stranded will become a single stranded after the ligase DNA was added.
The DNA ligase is used to repair back the breaks in the backbone in these two DNA molecules. So what happens is, this enzyme will form back the sticky end between these two DNA molecules based on complementary sequence. As a result, a combinant DNA was produced, and the cycle keep going on, reproduced in bacteria.
So, as a conclusion, this restriction enzyme is very important in findings of biotechnologies, such as recombinant DNA, DNA mapping, and many more. Well, that's it for today. Thank you for listening. Bye!